Hi all, thanks for joining me on this next presentation journey as I discuss the biosynthesis of cholesterol and the cholesterol-reducing drug lovastatin. I hope you enjoy it. Cholesterol is a lipid with a unique structure consisting of 4 fused hydrocarbon rings. Cholesterol is present in most animal membranes in varying amounts, but is absent in prokaryotes and intracellular membranes. Its hydroxyl group forms hydrogen bonds with the carbonyl oxygen of a phospholipid head group, while the hydrocarbon tail positions itself in the nonpolar core of the bilayer. Since the structure of cholesterol differs from phospholipids, it disrupts the normal reactions between fatty acid chains and can insert itself into bilayers perpendicular to the membrane plane. As such, it is a key regulator of membrane fluidity. Cholesterol also forms lipid rafts, which result in membranes that are less subject to phase transitions. This characteristic increases the permeability of the cell membrane to hydrogen and sodium ions. Although cholesterol has a dubious reputation in contributing to coronary artery disease, it serves a variety of essential functions in the human body. This includes synthesizing steroids, vitamin D, and sex hormones, which control a myriad of bodily functions. Assisting the liver in manufacturing bile acids, essential for digestion and absorption of fat-soluble vitamins such as vitamin A, D, E, and K, forming the myelin sheath, a neuron consisting of fat-containing cells that insulate the axon from electrical activity, as a cell to interconnect lipid molecules which are needed to stabilize our cell membranes, maintaining body temperature, protecting internal organs, and modulating the fluidity of cell membranes. Cholesterol is synthesized by virtually all tissues in humans, although the liver, intestines, adrenal cortex, reproductive organs, and placenta make the largest contributions to the body's cholesterol pool. Cholesterol is an essential molecule. However, cells can synthesize it from simple precursors, so dietary sources are not necessary. Cholesterol is a 27-carbon containing compound. All the carbon atoms in the cholesterol is provided by acetate. NADPH provides the reducing equivalents. The biosynthesis pathway of cholesterol is highly endergonic and requires ATP. To produce 1 mole of cholesterol, 18 moles of acetyl-CoA, 36 moles of ATP, and 16 moles of NADPH are required. As we have learned, cholesterol is synthesized from cytosolic acetyl-CoA by a sequence of reactions in the same way acetyl-CoA is produced from glucose. Cytosolic acetyl-CoA forms acetoacetyl-CoA, which condenses with another acetyl-CoA to form HMG-CoA. Cytosolic HMG-CoA, a key intermediate in cholesterol biosynthesis, is reduced in the endoplasmic reticulum to mevalonic acid by the regulatory enzyme HMG-CoA reductase. Mevalonic acid is phosphorylated and decarboxylated to form the 5-carbon isoprenoid isopentyl pyrophosphate. 2 isopentyl pyrophosphate units condense, forming a C10 compound, geranyl pyrophosphate, which reacts with another C5 unit to form a C15 compound, farnesyl pyrophosphate. Squalene is formed from 2 C15 units and then oxidized and cyclized, forming lanosterol. Lanosterol is converted to cholesterol in a series of steps. The ring structure of cholesterol cannot be degraded in the body. The bile salts in the feces are the major form in which the steroid nucleus is excreted. The conversion of HMG-CoA to mevalonate by HMG-CoA reductase is the rate-limiting step of cholesterol biosynthesis and is under strict regulatory control. As a result, HMG-CoA reductase is a popular target of a class of cholesterol-lowering drugs called statins. Lovastatin is a potent drug used for lowering blood cholesterol. Like other statins in its class, it acts by competitively inhibiting the enzyme 3-hydroxy-3-methylglutaryl coenzyme A reductase, involved in the biosynthesis of cholesterol. Lovastatin is synthesized through the process of fermentation. Its mechanism of action is simple. When the lactone ring of lovastatin is in its open form, as it would be in the human liver, the structure bears a strong similarity to HMG-CoA. As a result, it is a very specific competitive inhibitor of HMG-CoA. An attractive characteristic of lovastatin is that it selectively reduces levels of LDL while leaving HDL unaffected, or in some cases increases its levels. So how exactly do statins work? Statins act by competitively inhibiting HMG-CoA reductase, the first and key rate-limiting enzyme of the cholesterol biosynthetic pathway. Statins mimic the natural substrate molecule HMG-CoA and compete for binding to the HMG-CR enzyme. This competition slows the rate of mevalonate production, the next molecule in the serial steps to produce cholesterol. Hence, in the presence of statins, the precursor HMG-CoA is not efficiently processed forward to produce mevalonate, blocking the pathway. The molecules of statins vary from one another in their ring structure and chemical side groups. These differences affect the pharmacological properties of each statin by varying the affinity to the active site of the target enzyme, HMG-CR, the rates of entry into liver versus non-liver cells, the systemic availability of the compounds in the body, and the biochemical metabolism and excretion pathways that affect the biologic half-life of the active compound. Let's talk a little about good and bad cholesterol and what that really means. Cholesterol and triglycerides cannot dissolve in the blood. They must be transported within the cells by carriers called lipoproteins. Low-density lipoprotein, LDL, and very low-density lipoprotein, VLDL, are known as the bad cholesterols, while high-density lipoprotein, HDL, is known as good cholesterol. Hypercholesterolemia is a condition when there is an extremely high level of cholesterol in the body. Usually this means that there is a high concentration of LDL and low concentration of HDL. When too much LDL circulates the blood cells, it can build up on inner walls of arteries that feed the heart and brain, and therefore cause clogging of the arteries. HDL is known as good cholesterol in that it removes excess cholesterol in the arteries and transports it back to the liver for excretion or reutilization, and thus prevents the arteries from clogging. That wraps up my presentation on cholesterol and lovastatin. I hope you found it informative and helpful. I look forward to watching your presentations as well. Have a great weekend.